Thank you all for attending the Lupus Foundation of Minnesota's webinar on Update in Lupus Research. We appreciate you all attending. And today we have Dr. Timothy Niewold with us. He's an associate professor at the Mayo Clinic, Division of Rheumatology. My name is Catherine Taco, and I will be moderating the webinar. Just wanted to point out a few types of functionality that you have available to you as a webinar participant. You'll see a toolbar, and there is an orange rectangle with an arrow in it. That can help you, if you want to see a full screen, that can hide your toolbar for you, and you'd be able to see the presentation screen in its entirety. And Dr. Niewold has invited us to ask questions throughout the presentation today. And the way you can do that is just at the lower right-hand side of your toolbar, there is a place where you can type in a question. It might say question or chat. And you'll be able to type that in and enter it, and I'll be able to see your question. And when time is appropriate, I can have Dr. Niewold pause, and we'd be able to address that question at the time. All right, and it does look like we have a majority of our participants signed in and ready to go. One reminder that I also wanted to let you know is that we are recording this webinar, and it will be available on the Lupus Foundation website for future viewing. So now I will hand it over to Dr. Timothy Niewold. Thank you.
Okay, great. And I'd like to thank the Lupus Foundation of Minnesota for inviting me. As you all know, the Lupus Foundation of Minnesota has been big supporters of lupus research. And myself, I see patients, and I also run a lab directed at understanding lupus from the sort of biological or immunological perspective. And so today I just wanted to run through some updates in lupus and try to, I think we've got a broad audience, which is great, so we're going to try to explain and put into context some recent findings and trying to understand some of the causes and triggers of lupus. Next slide. Okay. So the first slide here about lupus, and probably most of you are very familiar with lupus yourself, but I'd like to do at least a little bit of introduction and give some perspectives from, I think, the point of view of both, people trying to treat the disease or solve it with research. It's sometimes called a prototype autoimmune disease, although the manifestations are fairly different between different people. And so, that's what I mean by protean, is that it's called a prototype, but I'm not sure how well we understand this prototype of autoimmunity. And so we do, all lines of evidence point to the idea that it's an abnormal activation of the immune system that causes the disease. But when you start asking more questions, then we quickly realize we don't have a lot of the answers. Some of the things we know from following patients over time is that it does have a predilection for inflammation in the skin and the joints and the kidney, of course, and then there are also a lot of blood count abnormalities that can come with lupus. And then the course of the disease is highly variable, and it tends to be a condition of both flares and remissions. And so this also makes it somewhat challenging to treat and to use the right amount of medication at the right time. As many of you know who either have been affected by lupus yourself or treat patients with lupus, the current state-of-the-art therapy is suppressing the immune system, and so we'd love to get more specific medications, maybe ones that could really get to the heart of the condition a little bit better, because many of the things that we currently use bring down the activity of the immune system in a fairly broad or nonspecific way. The last bullet on the slide, I think, sort of brings me to the idea, behind, I think, many people's research into lupus, is that if we can understand better the causes of lupus, then that might help us direct our therapeutic efforts, and maybe that's where we can make a dent and understand better what we need to treat. Next slide. Okay. And these are a few of the pictures from the American College of Rheumatology slide collection of lupus, as far as showing the some of the common manifestations. So in the upper left corner, there's a picture of the classical malar rash, and that's something that many people are affected with and is fairly diagnostic. Tends to affect both of the cheeks and then spare the nasolabial fold, and very characteristic. If you start asking why does the rash hit there and not anywhere else, we really don't know, honestly. And so so there are a lot of interesting questions, and I think from the perspective of research, there are so many things we don't know. In the right across in the upper right are a couple pictures from this is slides made from kidney biopsies, and it's showing some of the characteristic patterns of lupus when it affects the kidneys. And then down at the bottom, there's a rash on the hands. In the lower left, there's a rash on the hands that occurs between the knuckles and not over the knuckles. Again, really characteristic as a location, but interesting, and again, why it goes there and not elsewhere, we don't know. And then sometimes the arthritis can be really severe, and so luckily that's not common, but deformity of the hand, as shown in the other picture, can happen in some cases. So it's a really severe disease, and it's one where we're hoping to get better treatments for. And I think all of us who've either been personally affected or taken care of patients with lupus, we appreciate just how bad an actor it can be. Next slide. Okay, so one of the ways that we've thought of to try to figure out what causes lupus is by trying to work on the genetics of lupus. So we've known for a long time that lupus tends to run in families, and so that would suggest genetic causes. And so what we think, I think the big picture idea is that if you could find the genes that cause lupus, then maybe that would tell you what particular molecules or what particular immune system problems you might want to intervene on. There are a lot of different things, a lot of different parts of the immune system, and we have 20,000 genes, so having some sense of which one of those particular features are underlying lupus would be very helpful. But that brings up the question on this slide, I think, that I wanted to hit on as well, is why does this make sense? So this is a question that we get quite a lot, is why would our own genes give us lupus? And that it doesn't sound like a nice thing. Why would we have genes that would give us lupus? I think it's probably getting too much of a good thing, essentially. So in this slide, we're making the point that for most of human history, we've been killed off largely by infections. And most likely, the differences between our ancestors surviving infection and not surviving, there were probably some genetic factors that helped turn on the immune system more aggressively, or were high functioning within the immune system. And as time goes on and we go through repeated bouts of this, we'll tend to enrich our population for these immune defenses. It's possible that once you start making enough of these genes that turn on the immune system, it's always possible that someone may sort of have bad luck and inherit too many of these, or maybe you get bad luck and you inherit 5 of them that all work in a very similar way, and they all really compound each other in one particular area in the immune system, resulting in a turn on of the immune system that can't be turned back off. So in general, we think that many of the genes that are found to be associated with lupus are actually pretty common in the population, and we think that they are there because they actually perform some useful function in immunity, probably, but then in combinations they may actually cause us some trouble. And I think it's this arms race against microbial infections that results in this. We're always under pressure to develop a stronger and stronger immune system, and then there's always some chance that as the genes are passed down through the generation, that someone may, by bad luck, kind of get too much of a good thing. And the precedent for this is fairly strong. I think one of the examples I can point out is the sickle cell gene. So Some of you may be familiar with this gene. It causes a change in our red blood cells that makes them in the shape of a sickle, which is where the name comes from. And that can cause problems in the small vessels in the circulation. These sickled cells can sometimes cause kind of traffic jams in the blood vessels because they're not normal shaped. And so that sounds like it might not be a good thing, but the reason it's been transmitted in human populations is that it actually protects you from malaria. And so you find this gene variant exactly in the areas where malaria lives. And so it's one of these really strong examples of how infections and the sort of microbial world has actually changed our own genome and had an impact on us. And I think this probably happens in many other ways as well, which we may not always know exactly which infection pairs up with which gene. And we can either do next slide or if there are any questions.
Okay, I will advance to the next slide, but just as a reminder, you can type in questions as you have them. Oops.
I may not always be clear, so feel free to ask questions. I guess I referenced this idea a little bit on the previous slide too, is that probably most times lupus is caused by many genes coming together in a given person. There are some rare familial cases where it runs really strong in a family and every single generation is affected, and we found a few single gene variations that seem like they can cause lupus almost all by themselves. But that's the exception. That is probably less than 1% of the lupus community. If you took all lupus patients, it would probably be 1% or less that has a single gene that causes the condition. So we think that it's a multifactorial cause, and that's one of the things that makes it complicated when we try to study it. There are some models in biology, it goes all the way back to Gregor Mendel and the pea plants, where you get one gene and then you get one thing that comes with that gene. When things behave that way, it can be a lot easier to figure out. When it takes multiple different factors to all come together and cooperate, then it can be fairly hard to figure out. But we've been working on it and making progress. And it seems that a lot of our autoimmune diseases, actually, not just lupus, but also rheumatoid arthritis, multiple sclerosis, and type 1 diabetes seem to behave in this way, where we don't find one single gene, we find a bunch of genes related. And so we think as far as the biology, that probably makes sense, and that maybe it takes more than one thing to turn on a particular switch in the immune system, or to make something happen. And so that's what we've been finding. Next slide. And I just put this in. This is so for scientists in the audience. This is my favorite paper from 1918. And if you're going to read one paper from 1918, I would probably read this one, although I'm kind of biased in liking papers about genetics. But this is a very landmark paper in genetics, and it got to this idea that probably many of the things that occur in people and even in other in other species are contributed to by multiple different genes. And so in the early 1900s, there's this debate, and I guess it still kind of goes on now, between Mendel's genetics, which is basically one gene results in one finding. Either a green, one gene may give you green peas, another one give you yellow peas, and that makes sense because we think the genes all do something individually by themselves. When you study something like human height, which is what this paper based a lot of its data upon, it's not so simple. And many of us can find this even within our own families, or other families that we know. You can sometimes have two tall parents and a short child, or the other way around, you could have two short parents and a tall child. But on average, when large studies were done of human height within families, it was found that on average you're more likely to have a height that resembles your parents. And so, so that didn't fit at all with Gregor Mendel's models and the single gene model. And so, so this paper basically comes up with one of the, I'd say, fairly seminal solutions to the problem. the answer is that basically everyone is right, and so many times that's the case in science, is that everyone is actually correct. So so if you allow for, say, human height to be determined by multiple different genes, with each gene being inherited just as Mendel said it would be, then everything works fine, and pretty quickly you get sort of a bell curve shaped distribution where, if there are 20 genes involved with being tall, it's very unlikely that anyone's going to get all 20 of them, and maybe most people will get somewhere around 10, and then most people will have a fairly average height. Likewise, it's very unlikely that you would not get any one of those 20 different gene variants, and so therefore there'd be very few people that would be 0 out of 20 that would tend to be very short. And so we think that a lot of biological systems and also some of the autoimmune conditions and diseases that we see are inherited in this way. And I just presented this as an example. This paper's on JSTOR if anyone's interested, and it's sort of a nice reconciliation of and lead into modern genetics. Next slide.
Okay, we actually have two questions, Dr. Niewold.
Yeah, that's good.
One is, since it's been 10 years since the human genome map was completed, how close are we to finding improved treatments, and how much did that accelerate the progress, or didn't it?
So that's a great question, because I think that's a lot of the promise of genetics and the human genome, that we sort of unravel this, and get down to the heart of many things. And so I think we're getting there. I can tell you the updates after the Human Genome Project. A couple big projects have occurred. So the Human Genome Project aimed to get sort of the basic roadmap, of our human chromosomes. But it was only a couple people, and so it's fascinating. A couple things you should know is that as a human species, we're all very, very close to each other genetically. So we're all very new on the planet, and for probably most of human history, there weren't too many of us until recently. We've kind of bloomed across the whole planet, we've become very successful as a species. So go us, but it also means that we're very, very related to each other. And so you can actually sequence just a couple humans and get a pretty complete map, because we're, I think compared to other species like some of the bird species or other things, we're almost photocopies of each other. But there are still variations between people. And so, if 99% is the same, there are also variations. And so in the 10 years after the Genome Project, one of the main efforts was to map the differences between people. So the Human Genome Project kind of resulted in one, this is our human sequence. And then the subsequent efforts have been designed at detecting variations. And those are still ongoing. To get to, so I don't get too far off your question, the variations we think are probably going to be pretty interesting with respect to disease, and because, luckily not all humans get any one condition, and we think that if the genes are related to getting a condition, it should be that, some people maybe have that variation and some people don't, and then some people are more likely than others to get that particular condition. So the mapping of variation has come quite a long ways, and I'll say that where we are, in drug development is not so far along, but where we are in at least finding the variations linked to lupus is getting better. So I think it's hard to point to an example within autoimmunity where the genetics has led to a brand new drug that we maybe wouldn't have gotten to otherwise, although in cancer, this has happened a couple times now, and so I think it's not out of the picture. There have been a couple new cancer drugs that have been developed for patients with a very specific genotype, where they can genotype the person and then be able to tell that a particular drug is going to work very well on them. So I'm hopeful. It'd be hard to predict exactly how long it'll take us, but I think at least there are some examples there.
Okay, thank you.
That answers the question. I guess it's hard to give immediate feedback, but type something in if I didn't get to it, if I didn't answer anything you wanted. And then you said there was another question?
Yes. What specific environmental factors has research found impact lupus onset or flares?
So that's a great question too. And this talk is actually biased a little bit toward genetics, probably because we've had much more progress in genetics, and some of our scientific methods for genotyping, it's one of these growth areas. It's like a tech growth field where it grows logarithmically year after year. How much genetic information we can get, and so you'll see people, and I'm probably balled up in this group of people where they'll just talk about genetics and not talk about the environment very much. The problem is that it is kind of hard to study environment, actually. And so that's why, when you hear scientists talk, they won't always talk a lot about it because they get worried that they haven't got too much to say. Things that we know, though, Epstein-Barr virus has been linked to lupus. It's a tricky relationship because this is a very common virus in the human population, and probably greater than 90% of us are exposed to this and have some kind of a infection at some point in our life. We think it probably can cause a syndrome kind of like mono, but many people are probably infected and don't even really know it, or they have kind of a viral syndrome, who knows, and for a couple weeks and you get better and you never get checked out. So this is why I say it's a little complicated, that if most people are exposed, and then luckily we don't have 90% of the population with lupus. So there must be background factors that help that particular environmental factor work, and I think in general we think not so much in stark terms of genes versus environment, but almost more in cooperation, probably genes plus environment, where you get the right environmental trigger on the right background, and then that can cause trouble. And that's one of the few environmental factors that we know of as far as causing lupus. The second part of your question, as far as causing flare, I think we know a little more about that. So UV light is a pretty typical cause of flare in lupus, and that's one that we've known for quite some time. And if there's anyone on the call who's been personally affected by lupus, you've probably heard your physician tell you to avoid UV light, and use sunblock and everything. Viral infections seem to trigger lupus as well, so even things just like a simple common cold can sometimes trigger a lupus flare-up. We don't necessarily think, besides this Epstein-Barr virus connection, we don't think it's a general thing that all viruses necessarily cause you to get lupus, but if you already have lupus and then you get infections, either viral or otherwise, that can sometimes cause a flare-up. And that's also, it's still unpredictable because people with lupus will sometimes get a virus and be fine, and then sometimes get a flare-up. But those are some of the best established environmental factors. Okay,
thank you. And I will continue forward in your presentation there.
Okay. So this came up a little bit in my answer to the human genome question, is that I think we do feel some feeling of triumph in genetics, in that we've actually got some of the genes nailed that cause lupus. And there are quite a few now. This slide is really technical in the picture, but the title is really the take-home point. And this was even a paper from 2008, so it's getting to be an older paper now as far as science goes. And they had hit 6 different genes, and I mean we've had a number of subsequent studies where we've actually got the list up to about 30. And this is all fairly recent. If you go back even to 2004, 2005, we maybe had 1 or 2 genes implicated in lupus. So we've had a large expansion of our knowledge in this regard, and I think it's a very hopeful time for many of us, where we feel like we can stop complaining that we have no idea about what causes lupus anymore from the genetic perspective. Now we've actually got a list generated. Next slide. And this is just a one of the lists. This one's actually getting a little bit out of date too, because it's from back in 2010. But I put this slide in, and again, if you have a medical or science background, you might be interested in some of the list of potential functions there on the right side of the slide. And the summary is that basically many of the genes that we're finding have functions within the immune system, and so that fits with our idea of lupus as being an autoimmune condition. A lot of these were found by basically screening across the whole genome too, so it kind of gives us an extra level of confidence that these are probably, we're probably on the right track. It wasn't that, we decided to look at one thing and find out yes, no, but we actually just looked across the whole genome and let whatever come to the top, and then we ended up finding a lot of immune system genes. The odds ratio, again, just the background on this is that this is the kind of your fold risk of getting lupus if you carried any one of these particular genetic variations. It sounds a little bit scary, like the top one is a 2.4 odds ratio, meaning you would be 2.4 times more likely to get lupus. The other variable that you put in that equation, though, is the population rate of lupus is somewhere around 0.1 or 0.2% in the general population. So it's not completely rare, but it's also not a super common disease either. So if you just, say you got your 23andMe genetic report back and they said you carried the HLA type associated with lupus, that would mean that you would take that 0.2% times 2.4, and so you'd have about a 0.4% chance of getting lupus as opposed to 0.2% in your lifetime, just having known that one thing. So that's unfortunately the state of the art as far as prediction. Despite finding all of these genes, we're still not at a point where we can predict who's going to get disease and who's not based on genetics, because the odds ratios are not that high. A twofold risk when you're talking about a 0.1, 0.2% chance doesn't change that much. On an individual level, if that was your report after sending your genome off for one of these commercial sequencing things, you hopefully wouldn't lose any sleep over it, because you'd go from a 99.8% chance that you're not going to get lupus to a 99.6% chance that you're not going to get lupus. So there's a lot to learn. I guess it's both elation in that we're finding the genes, but the complexity, this goes back to the earlier slides, is that we think that it's going to be fairly complex and there will be a lot of genes involved. Something that we haven't been able to identify much yet, but we're hopeful, is that we may find genes that work together, and there may be synergies out there. And what I mean by that is you may find something that by itself has an odds ratio of 1.3, and maybe another one by itself has an odds ratio of 1.5, but maybe when they're both found together, it goes up to like an odds ratio of 20 or something like that. So if we found combinations like that made sense, then we might be getting closer to prediction. So we've been trying, and it's not for lack of effort, but it's just we may not have enough of the players known yet to be able to detect these. Either questions or next slide. Okay, so this is a very slight variation on the previous slide, but one of the things I wanted to get into at least a little bit, and those of you who followed the lupus research that's come out over the last 5 years or so have probably heard of this, but many of the genes that have come up in lupus have been linked to interferon. And so this has been a particular interest of us and many other groups actually that are studying lupus, is that it seems like interferon alpha is one of the immune system molecules that has been linked to lupus in many different ways, and it also genetically. So it seems that there's sort of a genetic link to making more interferon alpha, and so it ties in with evidence from many other sources that this is probably an important cause of lupus. Go to the next slide. Okay. So a little bit of background on interferons, because it's like many things in science, it's sort of old and then new again. We've known about the interferons in the immune system for quite some time, and so it's a family of molecules. There's interferon alpha and interferon beta. We think that alpha is probably more involved in lupus, but they're both normally involved in defending us against viruses. And so that gets back to the very early slides where we talk about, too much of a good thing, is that the, interferon alpha is something that we make ourselves and is part of our normal immune response, but we think it's sort of being made too much and not getting shut off very well in lupus, or maybe a combination of both. But we find sort of inappropriately high levels of this particular immune system molecule. We've known this for a long time, so we've known this since the 1970s. And I think some of the limits were in the technical methods as far as how you could measure it, and also being able to study large patient groups. There wasn't a lot of motion in the 1980s and 1990s on interferons, but it came to the forefront again in the early 2000s, and this was when we started doing these gene screens in the blood, actually. So you could really see that if you looked at the circulating blood cells from patients with lupus, this pathway was really turned on, and that it was one of the most dominant pathways in the circulating blood cells. And so that really, I think, put it back on everyone's radar in a big way, and stimulated much more work in the area. And this was around the time when we began to get interested again, too, as something where we might have, you know, an identifiable target in the condition. Next slide. Okay, and so one other sort of piece of evidence about interferon alpha, it's been used as a drug. It still is used as a drug, actually. So for us in the rheumatology and lupus community, that sounds like it's really probably not a good thing, but it is an antiviral molecule, and the current condition that's used most for is actually chronic viral hepatitis. And there, of course, the rationale is pretty straightforward, is that it's an antiviral, you know, molecule that stimulates the antiviral immune system, and so you might be able to boost someone's natural antivirus response. In some cases, giving interferon alpha to people has actually resulted in lupus. Luckily, it's not very often, otherwise it wouldn't be available as a therapeutic, but somewhere a little less than 1% of people receiving this medication may develop lupus. And it's actually one of the things that's on the package label and you have to watch for. The thing that makes it not so bad is that usually if you stop the interferon, then the lupus symptoms go away. For you know, us on the research side of things too, it's interesting in that it's kind of a human experiment, giving this drug as a virus treatment, because you know it's something that you would never do. You know, we wouldn't give interferon to people for no reason, but it's being used already, and then you can see this phenomena where people develop lupus when they get interferon, and then it goes away when you take it away. So it's interesting. It suggests that this is likely to be a causal molecule, and then the genetics would also suggest that this might be involved in the early stages or the causation phases in lupus. Next slide, or questions? Okay. So to push that a little farther, one of the studies that we had done was to look in families. And so we looked in families that had at least one person affected by lupus, and we measured the levels of interferon alpha not only in the person affected by lupus, but also in their relatives. And you could find a little bit of an echo, actually. As you can see on the graph, the people who had lupus had the highest interferon levels, and that was fairly striking. That's the big tall bar all the way to the left. But healthy first-degree relatives, so this means people in the nuclear family, you know, father, mother, brother, or sister, on average all had higher interferon levels than healthy unrelated people if you just took everyone on average. When you looked at individual families, it wasn't that everybody had that, but you would tend to find people who had high levels here and there within lupus families. So maybe it would be a mother or a father or a sister or a brother, you know, and then when you took an average of all the relatives, then you got an elevation. It wasn't as significant as the patients, but it was significantly higher than the unrelated individuals. So so this was, we did this study because we, you know, in studying causes in humans, it's a little trickier. You know, the people who study mice often do it because you can really manipulate a lot of factors, and you can control the environment, and you know, there are all these things you can do when you study mice. With people, you're limited more to observation, but we thought this would be an interesting observation if we looked at families, and this would be the same pattern you would expect in an inherited risk factor. And so we found it did tend to track in particular families. Some families didn't have high interferon at all, neither the patient nor the relatives nor anyone, and other families seem to be kind of higher interferon families where a number of different members had high levels of interferon. And that would be what you'd expect if there were some genetic factors that were tracking with this.
Okay, thank you, Dr. Niewold. We do have another question here.
Okay.
So, has it been well established that if alpha causes SLE, Or could it be that having SLE increases IF alpha?
So that's a great question, and that's actually about where, I mean, I think that's the case we are trying to make. So so in humans, you know, it's always tricky, but I think the last couple slides we've been kind of building the case that the interferon alpha comes first. So so you know, ordering things in humans, you can't just take a group of humans, you know, and manipulate the scenario, you know, like you would in mice. So what are some lines of evidence? So I've been trying to present some of these lines of evidence. The interferon used as a therapeutic was fairly striking because that's an experiment that you actually couldn't really do. But there you have someone who doesn't have lupus, and then they get interferon and get lupus, and then you take it away and the lupus goes away. So that's fairly striking in support of interferon causing lupus. This family study also would suggest that the interferon may be inherited and therefore may come first. I'll say that my own personal gravitation toward genetics relates a lot to this question, actually, from a participant, is that the genes that we are born with almost by definition come first, you know, and many of the other processes then could at least be ordered as second, you know, and that's, in many studies, it's often hard to tell the chicken and the egg. But when you do genetics, you know, we don't have any way we think that the interferon could change your genes, you know, and so therefore if you find a gene that's related to interferon, it should be because there's something about that gene that causes the interferon. So it's a great question because assigning the causation is always tricky. I'll say even in mouse studies, causation is always hard. You know, the best thing you can do usually is to establish multiple lines of evidence, and if they're all suggesting a similar thing, then you start to believe it. So I think with interferons, we've reached that threshold, at least in my own mind, that there are a number of things suggesting that this may be an early factor and a causal factor.
Okay, great. Thank you for taking that question, and feel free to submit additional questions if there are any for the group or by the group. So I'll forward the presentation.
Okay. And so this slide, I think I guess I maybe mentioned a little bit, but just to make it a little more clear, this goes back to some of the earlier slides, you know, the single gene inheritance versus multiple gene inheritance. It actually looks like even when you study something that's molecular, like interferon levels, and you study that in families, it wasn't a simple single gene thing. And it looked like there were, you know, it fit a model of having multiple different genetic factors, and it didn't fit any of the Gregor Mendel, you know, single gene models like the studies in the pea plants would have fit. So so that was, you know, I guess we probably should have expected that, and many, I think in the broad sense, if you're afraid of complexity, then studying lupus is not a good idea. So we're not, and we're not afraid of that, and so that was. But I just wanted to make that point. Actually, it's not a it's fairly complex, and all of the different patterns in the families were highly variable. So we didn't see any one particular family. It didn't always say go from mothers to daughters or fathers to sons or anything like that. It was just more likely if you were more closely related. And so that would suggest that multiple factors should be involved, and the factors could be, they don't have to be all genes. There could be genes and environmental factors that could be involved. Okay, next slide. And so from the gene side, this is just a recapitulation of that slide again. It would make some sense because there are genes that we've found that relate to you know, function within the interferon alpha pathway. We've actually gone on and studied a number of these in the lab and tried to determine if you carry these particular gene variants and you have lupus, do you tend to have higher interferon levels? And we've generally confirmed that. So we've found that many of these genes actually do correlate with having higher interferon levels in patients with lupus. So that's sort of fitting this model as well. You know, again, that's another line of evidence we've tried to develop that would support this idea that the genes are tuning the interferon response and having a causal role there. Next slide. Okay. This slide, and so I'm sorry if this looks a little scary, but it will go through. This is in trying to draw a cartoon of the immune system. This is one of these models, and if you've looked at some of the studies or other research presentations, this has been a major model that's been proposed in lupus about how do you get interferon produced. So the genes do seem to have an impact, and which genes you carry make you more or less likely to have high interferons. But one of the other really interesting stories is that the autoantibodies that we make in lupus, they probably stimulate the immune system in the same way that a virus might. And so that's what this cartoon is trying to show, is that we make these the antibodies stick to their targets, in circulation or in the tissues. And then when they're taken up by immune cells, they contain nucleic acid, which is DNA or RNA. It's one of the striking features of lupus is that you make antibodies against the nucleus of the cell, which is where our DNA is held, and that's where the anti-nuclear antibody test, that's where that comes from. We think that because these antibodies can bind nucleic acids, they may also be able to stimulate the immune system, and that they get taken up and they look like a little lump of protein and DNA, which is actually what a virus usually looks like too. Viruses aren't full living cells; they're actually particles that are just just the bare bones of a replicating organism, basically, where they have some proteins that let the virus get into cells, and then they have a little DNA that allows the cell to make more viral proteins. So, so we think that this is what's going on. This is a bunch of elegant work in this area to show that the antibodies that are made in lupus that we use when we diagnose lupus, like the anti-nuclear antibody and the double-strand DNA antibody, they do probably result in producing interferon when they're seen by the immune system. Could do the question or next slide. And this is true if you just look at large patient groups too. So so people did what you might think is that they took the antibodies and purified antibodies from lupus patients, and then they put those antibodies along with their targets on the immune cells, and you could see, in a culture dish basically, and you could see interferon being produced. We looked at large populations of lupus patients, and we tried to see, is this correlation true in groups of patients? And we did find that actually, was that some of the strongest links with having high levels of interferon were some of the typical autoantibodies that we measure either in diagnosis of lupus, like the Smith antibodies or the anti-Rho antibodies, or double-strand DNA antibodies, which we often follow. Actually, when we follow lupus patients, we'll follow the double-strand DNA antibody levels. And if you've been affected by lupus, your doc probably checks those many times when you come in. So those were some of the strongest correlations with having high interferons when you looked at the patient populations. And that was kind of exciting because it's sort of hard to measure interferon, and there isn't a laboratory test available in the clinic that you can order to measure interferon. But we are already getting a lot of information about these antibodies, so there may be tests that are already available that would give some prediction about whether a patient may be likely to have high interferon or not. To go to the next slide. And so this is another cartoon trying to describe this relationship. We think that the antibodies, probably complex to their target, and then this endosomal toll-like receptors thing in the middle, those are the receptors that normally recognize viruses. And so we think these antibody immune complexes are kind of fooling that system. And then this probably plays upon your genetics. So we do find that there are people who have these antibodies and they don't have high interferon levels. So like many things in medicine and in life, it's not 100% yes, no, it's some but not all. And so that's where we think probably the genetics comes in as well, is that if you have the antibodies and then you also have genetics that makes your interferon system sort of hyperactive, and this is where you could probably get into trouble because you've got a stimulus to the system and then you've got a hyperactive system. and then that might get beyond any of the normal control mechanisms and then result in high interferons in a constant way, and then risk of getting clinical lupus. Okay, go on to the next.
Okay, and just no additional questions at this time, Dr. Niewold, but just giving a time check, it's about 12:45, so approximately 15 minutes left of the webinar.
Okay.
So just giving you a heads up there, that's all. Thank you.
Okay, thanks. Yeah, I think we'll get through most of the slides, probably all, maybe at least most. Great questions, too, from the group, so I'm glad that we're doing those during the session. Mm-hmm. This slide is a fairly famous one from a paper in 2003, and I bring this up also for, this is for probably many of you are trying to think of the order of events. I think some of the questions have suggested this too. Does interferon come first or second, in disease? Is it that you get sick with lupus first and then get high interferon, or, that's one of those time sequence questions. And this was a study that addressed the time sequence of antibodies. And so this was really interesting, where they were able to obtain blood samples from people who were well when they donated blood, but then later on in life developed lupus. And so that could provide this sort of valuable window going backwards. In the medical clinics, we see people when they have symptoms, and it's probably already too late to get a sample before disease because it's people already coming with symptoms. But using these blood bank samples, they could actually look a number of years before. And so it looks like the autoantibodies come up years before diagnosis. And so it's kind of interesting because the genes, by definition, you're born with them, and then you start making antibodies, and you make more and more antibodies as time goes on. That's what the sort of rise in the line as it goes from left to right indicates. And then maybe that's when you sort of pass the threshold and make more interferon and then develop lupus. That's hypothesized, it's not proven, but that's sort of an interesting way that we think these things may come together. You can go to the next slide. Okay, and then this is just another slide kind of describing a similar thing where you could think of it almost like a two-hit model, where maybe your background genetics turns on your interferon pathway a little bit, and maybe that's not such a bad thing overall. Maybe you are less likely to get viruses. Maybe if you look at all of us together as a species, maybe it's good to have some of these high interferon genes circulating around. And that's probably why we still, they still persist in our population. Maybe the formation of lupus autoantibodies is one of the more dangerous things, where if you make these antibodies, then they chronically stimulate this interferon system, and maybe that's where you kind of break down all of the control mechanisms and start to get inappropriate immune system activation. Go to the next slide. Okay. And then, so this looks a little complicated too, and I'm sorry for that, but this is, I think, where, as a community, and definitely our group is interested in, is trying to put together, what can we learn? Are there relationships that we can put together from what we've learned from studying human populations? And I think that there are. Even just the structure of this slide reflects some of the things that we've found so far. When we've tried to link genes on the left all the way over to some of the things that happen in the clinic, like getting a rash or getting renal disease or things like that, it doesn't work very well. So we haven't found a lot of things where we find, this is the lupus kidney disease gene. When we've looked, we've actually come up pretty well empty, or pretty close to empty. What has been really strong is when we look at the genes impacting on parts of the immune system, like, for example, whether you make these different antibodies or whether you make high interferons. And so this model has been interesting, and we think, and this is sort of my best stab based on current knowledge of how we think things may come together, is that the genes impact upon the immune system, and then depending on how the immune system responds, then that results in the actual clinical parts of either getting a rash or muscle pains or kidney disease or things like that. And so it's not trivial. I think if we get a really good map, this is obviously fairly preliminary, but if we can make it better and better, we might actually get better ideas of where we can intervene strategically.
Okay, and we do have another question for you. Do
Great.
do interferon levels go up with flares?
People have been looking at that for a long time, and it looks like the answer is probably not. So there have been many studies of this. There was a hope that this might be a marker of flare. When we first found high interferon levels, it tended to be in people who were having a lot of things going on with their disease when they had high interferons. We've done more detailed studies where you really track people over time and you follow interferon levels over time, to try to really prove it. And those have not been conclusive. It looks like it's probably not necessarily the case. There may be some people, but as a whole, it doesn't look like it's a great marker of flare. Some of the reasons why, I think actually some of the things we presented may describe some of the reasons why. I think some of it is, actually related to your background genetics, and then some of it is probably also related to the particular autoantibodies. And some of those don't really change very much. Of course, your genes don't change, and then the antibody panels too. Actually, if you would recheck, say, your Smith antibody, if you had a positive value at diagnosis, rechecking it 5, even 10 years later, it's usually still there. So I think some of the things that result in high interferon are actually relatively stable. It does tend to, if you look at what's different between people who have high interferon and people who don't, there are more autoantibodies in people with higher interferon. Probably greater disease activity on the whole, but you can't really track a level and then be able to predict who's going to flare. Okay. Thank you for the next slide there.
Yeah, thank you.
This is, I think this is the last major area we're going to hit, but this is an area that's been very interesting too. So lupus in African Americans. Many of you may know that lupus is actually more common in African American ancestry than in European American ancestry, and it's actually probably more severe as well. If you look at kidney disease, there's more end-stage renal failure in people of African American ancestry. If you look at disease activity scores, even with pretty simple studies where you just look across your whole clinic and then separate people by reported ancestral backgrounds, you can find differences. So we don't know exactly why this is, but it seems that there could be biological factors underlying this. And I just wanted to summarize briefly some of the work in this area as well. You can do next slide. Okay, so genetics, I would say, is already suggesting that there are some differences. This is a paper from 2011, and I guess it's still the state of the art. We're just completing some of our first whole genome, across the whole genome screens in African American lupus, and we're actually expecting some results probably within about a year from this study. I think the genotyping is done and we're analyzing it now. But there's some genes that don't cross over. So some of the genes we found as risk factors in European ancestry actually don't cross over and result in any risk in African Americans. And so we think that as this Venn diagram is completed, there will probably be examples in the other direction too, where there may well be some genes that are particular to developing lupus in the African American chromosomes. Next slide. We looked at interferon levels, and so this was fairly interesting, and you can find higher levels of interferon basically in all of the non-European ancestry subjects. So in that table, that's actually the top factor on the table over there. It's not top because it was most strongly associated, but it was actually associated. If you had a non-European ancestral background, you're more likely to have high interferon levels. And then the graph over there is trying to demonstrate that with Asian American, African American, and Hispanic American subjects actually having higher interferon levels than European American subjects. And it's interesting because if you look at the risk of lupus and severity of lupus, both of those actually track in a similar way, in that non-European ancestry populations living in the U.S. tend to have more lupus and more severe lupus. And so it seems that interferon is tracking with that distinction. Do next slide. And when we looked at, we tried to sort of take, some of those factors and put them in statistical models together to see if we could figure out what are, what causes what, what's dependent upon other things and what's independent. And one of the things that came out of this, and this diagram is trying to show it, is that if you had some degree of African ancestry, you're more likely to have some of the particular autoantibody profiles, and then these were linked to having higher interferon. So it seemed that the link between African-American ancestry and interferon was somewhat indirect. If you kind of trace the arrows between the two on the diagram, you have to link through these anti-RNP antibodies to get there, suggesting that the ancestral background may have a bigger impact on the antibody profile, which then makes you more likely to have high interferon levels. We can do next slide, and I think this is basically another slide suggesting a similar thing from a recent study we've done, where if you separate it by antibody profile, the top EA stands for European American, and AA stands for African American, the bottom two panels, and then this RBP plus versus RBP minus indicates whether you have the antibodies on the plus side or you lack those antibodies on the negative side. And so the dots shaded red basically are all interferon-related genes that are turned on. So the more red is a greater turn-on of the interferon pathway. And so you could see in the European, if you just even step back a ways and just take in the red versus gray dots, you can see the top two panels in the European Americans, there isn't that much difference. You actually get a pretty strong turn-on of the interferon pathway with or without the antibodies, whereas in African-American subjects there was a really stark difference, and the antibodies were a really important factor actually in the turn-on of the interferon pathway. So, you know, the detailed molecular whys of this we don't know for sure, but we're trying to work backwards toward that. And I think it suggests something that we've probably suspected clinically for quite a while, is that, you know, the condition is, I don't think it's likely that it's going to be completely biologically identical across different ancestral backgrounds, and the differences in severity I think already kind of point us to that, and many of the clinicians, you know, feel like lupus behaves differently in different ancestral backgrounds. Do next slide. Okay, and this is a summary slide from this section, is that we do see differences in the type 1 interferon or interferon alpha pathway by ancestral background. So we see that antibodies are more important in African American subjects as far as turning on the pathway, and the antibodies are also more common in African American ancestral background. So you know, why does this make any difference? One of the ways is that we have drugs in trials now that target the interferon pathway, and so they'll probably be characterized by some patients that respond and some that don't, and so some of these factors may actually be useful in predicting who's likely to respond to some of these drugs and who may not be as likely. Okay, can do next slide. Okay, we're a little tight, but we're going to make it through, I think. So, and
yes.
There'll be a little time for questions for whoever wants to stay longer. I'm happy to stay on for a few minutes and answer questions too. So, you know, the conclusions, we think that interferon alpha is one of these causal factors, and it's probably inherited. Again, multiple lines of evidence we've tried to use to support that, but the idea that if you carry different genes, you're more likely to have high interferon, and then the clustering of interferon levels within families would also suggest a potential genetic cause. And we find many of the risk genes, you know, if you just look at the list of genes that have been linked to lupus, many of them have a role in the interferon pathway. You know, interesting story about the autoantibodies. Some of you that have followed lupus research for a long time, there is a sort of historical argument of whether the antibodies are causing someone to be sick. Are they you know, a bad actor, or are they just markers? You know, are the antibodies that we test, are they the fire or are they the smoke? You know, and I think in particular with the interferon story, that would build the case that they're at least part of the fire probably, if they're turning on the interferon pathway and if that's causing some of the disease to occur. Next slide. Okay. And then there's some differences, I think, in the way that lupus pathogenesis is going to occur between ancestral backgrounds, and this will be very important as we move into therapeutic avenues and trying to get the right medicine for the right person. And so we've been very active in trying to diversify the studies that are done in lupus from the biological perspective, and I think the clinical trials groups are doing the same. Groups that are not studied may or may not benefit from new developments, and so it's really important that we have a large and diverse population that we study when we do basic science or when we do clinical trials or any of these things, because we won't know unless we look. And then anti-interferon therapies are promising. That nothing's approved, but there are some in trials, and I think that for some of them it looks like there's a chance they may make it. It's always hard developing drugs, and some of them have looked good and then not made it, so we don't really know. But at least I think the biology of interferon is interesting as a potential target, and we'll hopefully see in the next few years whether these drugs are going to work or not.
Okay.
I think it's a thank you slide after that, right?
Yeah. Yes, there is, and we do have one question if you have time to take it, Dr. Niewold.
Yeah.
Yeah. Can you comment on Benlysta treatment? It's Benlysta.
Yeah. So that is that's very topical with that very last point about, and I was mentioning about drug therapy. So Benlysta is the first drug that's been approved by the FDA for treatment of lupus in approximately 50 years. So that's been the sort of sad history of lupus drug development, is that we haven't had the explosion that some other areas have had. In rheumatoid arthritis, there's been a big explosion of drugs, and you see the ads on TV and in magazines everywhere. Lupus hasn't experienced that, but Benlysta is one exception where we've had a drug that made it all the way through trials and worked. Some of the issues around using it have been that the trials were very large and they were well done, but the benefit of the drug was not huge. And so it was not nothing, but it wasn't a really huge difference. And so it's been hard to know where to put it in the therapeutic program. We definitely use it, although we don't use it first line, because I think we have some more faith in many of our old tried and true medicine. But it's nice to have another tool in the toolbox. And we do use it sometimes either in combination with some of the older medicines or sometimes by itself when older things fail. And so that's kind of where it is now. It's ridden a huge wave of excitement and then a huge wave of depression, actually, where everyone was excited there was a new drug, and then when they looked at the trial data, they thought, well, but it's not as strong as we would love it to be. And now I think hopefully we're shaking out somewhere in the middle, because it's definitely going to be useful for some people, and I think the challenge for us is to figure out just exactly who that is, and then maybe we'll have better success rates, if we can figure out which group of people within lupus responds. It's clear that it doesn't cure everybody with lupus by any means, but if there's some groups that respond particularly well, if we could identify those, then I think that would help us use it better.
Okay. And yes, we have no additional questions at this time, and like you said, there is a thank you slide here, and the Lupus Foundation definitely wants to thank you for your willingness to lead our webinar and the time that you've taken to put together the presentation.
Yeah, well, thanks for everyone who participated, and really great questions. I don't get a lot of feedback after my answers. I hope the answers were okay, because they were really good questions, and I know you didn't get a lot of chance to feedback afterwards after you asked the question, but it was that was great. It was very interactive, and I hope that everyone enjoyed it.
Okay, well, we'll definitely be looking for additional research findings that you have, and hope to have you back again.
Okay, great.
Thank you very much.
Okay, thanks everyone.
Mm-hmm.